My name is Chen Su, and I'm a graduate student working with Dr. Amin Imad at McGill University. Today, I am excited to share my research on multimodal inference of phenotype-relevant gene regulatory networks in human endoderm formation using inference chip. Human embryonic development is a complex process that lasts about 9 months, from fertilization to birth. However, most tissues and cell types are determined within the first few weeks post-conception. After fertilization, the zygote begins to divide and form a blastocyst, and the innermost layer of the blastocyst gives rise to 3 germ layers: ectoderm, endoderm, and mesoderm. Each germ layer has a specific fate, with ectoderm forming the nervous system, endoderm forming the digestive system and internal organs like liver, and the mesoderm forming muscles and connective tissues. Understanding the molecular mechanism behind germ layer differentiation is crucial for studying developmental disorders and disease. The formation of different cell types during endoderm development is driven by the activities of TRNs, which consists of regulatory relationship between transcription factors, TFs, and genes. TFs are proteins that can bind specific DNA sequence to control gene transcription. Reconstructing TRNs is essential in understanding the regulatory mechanism in this process and their potential links to common pregnancy and birth disorders. Many methods have been developed to reconstruct TRNs, but most of them do not relate TRNs to the variation in the phenotypic labels or scores, making it difficult to identify phenotype-relevant TRNs. To illustrate this challenge, let's consider an example that involves 2 conditions, case and control. In both conditions, there may be a linear correlation between the expression of a gene and the TF across all samples. However, the relationship in one condition, as shown in Figure A, may not be relevant to the phenotype, while the relationship in the other condition, shown in Figure B, is likely to be phenotype relevant. To address this issue, our lab has developed a computational algorithm called Inferno, which incorporates gene expression and phenotypic labels of score in TRN reconstruction. Building on our previous work, we have expanded Inferno to incorporate ChIP-seq data, allowing us to study the endoderm formation. Our novel Inferno chip algorithm integrates RNA-seq data from 3 independent studies, ChIP-seq data, and the phenotypic labels to reconstruct a phenotype-relevant TRN. The algorithm employs a probabilistic graphical model to combine regulatory evidence between TFs and genes with phenotype-relevant information of genes. Let's take a closer look at the pipeline of Inferno chip. In step 1, we perform differential expression analysis on the integrated gene expression datasets to obtain p-values that reflect how much a gene is differentially expressed in human embryonic stem cells versus endodermal cells. In step 2 and 3, we obtain 2 sets of p-values of TF gene associations from ChIP-seq and RNA-seq data. Those 3 sets of p-values are then fed into step 4 of Inferno chip, a carefully designed PGM that models the conditional distribution of those summary statistics and generates an initial bipartite graph. Finally, we perform several normalization steps and comparisons with different controls to identify TF gene relationships that are associated with the differentiation process. The PGM in step 4 is the core of Inferno chip, which I will describe in details. The goal of the PGM is to model the biological hypothesis about how each element, expression of the TF, expression of genes, phenotypic variations, influence one another. To design a PGM, we first define a binary random variable Tij for each path in the bipartite graph. Intuitively, if Tij equals to 1, it means TFI is regulating gene J, and gene J's expression is associated with variation in the phenotypic labels. On the other hand, if Tij equals to 0, it means at least one of those 2 statements is incorrect, which falls within case 2 to 4 here. The TRN reconstruction problems can then be formulated as obtaining a posterior probability on each Tij. After that, we define 3 sets of observed variables in the PGM. Pj is a random variable that shows the significance of the association between every gene and its phenotypic label. Pi-ij is a random variable that shows the significance of gene TF association from RNA-seq data while considering the effects of all other TFs. Qij is a random variable that shows the significance of gene TF association from the binding peaks of TF ChIP-seq data. We then use a causal directed acyclic graph to model the conditional dependencies between those observed variables and our predefined latent variable Tij. We run Markov chain Monte Carlo sampling to generate posterior samples of the latent variable Tij and form an initial graph. In terms of how we model the conditional distribution of each variable, we will cover it more in our poster session. As an example, the conditional distribution of each pj, which represents the gene phenotype association, can be modeled as either uniform or beta distribution depending on the value of Tij. As the first evaluation, we compare TF gene ages identified by our algorithm with GTRD database, which provides an aggregated TRN based on integration of many different studies. While this TRN is not phenotype relevant, we expect to see an enrichment with regards to ages we have identified, which we observe with the p-values of 2 to the power of minus 12. After that, we ranked TFs based on the numbers of target genes in the inferred TRN. Such TFs are expected to be important in the course of endoderm formation. Consistent with our expectations, we found various literature evidence for the roles of those TFs. Next, we use this experimental study as our primary data to further validate TF gene phenotype relationships identified in the TRN. The experimental study first used RNA-seq and ATAC-seq data to investigate dynamics in chromatin accessibility during the differentiation of embryonic stem cells to endoderm. Then, they performed perturb-seq screening to define the loss of function phenotype of potential molecular drivers in endoderm development. 3 TFs in our study was shared with this study. Focusing on those 3 TFs, we found that in all cases, we have observed a significant enrichment in the target genes identified by inferno chip and the target genes experimentally identified by this study. This shows that genes identified by inferno chip with respect to 3 TFs are significantly enriched for marker genes associated with early endoderm developmental process in the perturb-seq study. Thank you for listening, and I hope you have enjoyed it.